So welcome to your lesson on the cell membrane. It's called the fluid mosaic model because a mosaic is like having a million different tiles and it forms a picture. So it's many different components making up the one thing. It's called fluid because you can poke it and it moves. It's not a stiff, rigid structure. You can poke it and the little pieces will bounce down and up. Cell membrane is more than just the little line that we told you about back in grade 10. It's actually a really amazing, important structure. So we're going to focus in on what it does. And then you're going to do a whole set of labs when we do the mega lab that's going to be examining how we transport things across this structure. We'll have a formal lesson on transportation within the cell next. So first thing that you need to know is that every single cell is actually bathed in a liquid. It's not a dry cell. In fact, if it was dry, you wouldn't be able to breathe as a cell. You need to be able to have the oxygen and the carbon dioxide dissolved in a liquid in order to transport it across a membrane. It's the same thing that happens within your lungs. That's why when you breathe out, you see all that moisture, especially in the winter, we see your breath. You always have that liquid content vapor down in your lungs in order to make sure that the lining of your lungs is wet, not soaking wet, not full up with water. That would cause you more troubles, but wet so that the oxygen that you breathe in dissolves in the liquid. It can be transported across the cells and dumped into the blood where there's more water in there and that's able to carry and dissolve these gases. So you must have every cell bathed in a liquid in order to transport things properly between cells. We call that fancy cell liquid extracellular fluid when it's on the outside. Extra just meaning outside, right? Or beyond. Extra is beyond. So beyond the cell, we have fluid. So we have ECF. What type of stuff is in the ECF? Obviously water, and that's going to act as the solvent. So that's the thing that stuff is dissolved into, like molecules, like ions that we need, like those gases and whatnot, such as oxygen and carbon dioxide. You're also going to have these types of ions. These are going to be important for balancing the pH of the blood. It's going to be important for nerve transmissions and muscles and being able to operate little pumps that are going to be working within the cell in order to put things in and out of the cell. We're going to talk about these guys in more detail when we get to our homeostasis unit. It's also carrying a bunch of organic compounds like the vitamins that we saw before, like the macromolecules that you've been studying. They're floating around in there. And then as well, we have hormones, which are the chemical messengers. Hormones can be made from fats. You can have steroid hormones, or they can be made from proteins. Either way, they are produced by one type of cell. They get dumped into the blood, and then they have to go and influence the tissues of another organ. So they're going to have to be able to float there and get interacting with them. The ECF's job is also to carry away waste from the cell, so the cell can dump stuff out into the ECF, and then it gets cleaned up. And it's going to be getting rid of things specifically like ammonia. The ammonia is produced when you are breaking up proteins or breaking down nucleic acids. You're processing them. When you're processing or producing energy from molecules, we call that metabolism. So nitrogen forms when the ammonia, which is the NH3, is produced from breaking down the proteins. Ammonia is actually quite toxic to your body. It's going to be part of your liver's job to take that ammonia, takes a couple of them, takes a carbon dioxide molecule, shoves it together in a chemical reaction, and then releases it as a compound called urea. Urea then floats in the blood down to the kidneys and is removed in the urine. Urea, urine. It's the part that makes the pee yellow. It's the salt that you're trying to get rid of. And that's, again, is formed by trying to get rid of toxic ammonia. I think something like 100,000 times less toxic. It's in the urea molecule. So that's the liver's job. So let's look at the actual structure itself. And here in this diagram, you can see that there's a lot more going on than just thin line around a picture. Here's the phospholipids that we mentioned the other day. We've got a bilayer because there's two different layers of phospholipids. Why is the round part sticking up and the little feet sticking down? It's hydrophobic is when you don't want to be near water, and hydrophilic is when you do. So here we have the phosphate head. Remember, we have a glycerol molecule with a phosphate attached. The phosphate is charged, so it's polar. So it's attracted to water that's on the outside. The ECF has water in it. Then you have the tails that are these hydrocarbon fatty acid chains. And because of that, they are nonpolar overall. They don't like water. They're going to want to push away from water. And that's what forms the double layer, because these heads are also attracted to the water on the inside of the cell, in the cytoplasm. These heads are attracted to the water outside, and then the tails both go to the inside in order to away from the water. Hydrophobic tails, hydrophilic heads. So aside from that thin membrane, and that thin membrane, by the way, is super, super thin. It's about 7 nanometers. So on the micro scale, when you're looking through a microscope, you actually can't see the cell membrane. All you can see is the boundary where the cell ends. So all this time that you've been looking through the microscope, anytime you have, and you said, oh, there's the cell membrane, you can't actually see the cell membrane. You can just see where the cell ends because it interacts with light differently than whatever liquid you're floating your cell in. You'll see this when we do our lab as well. In the mega lab, we'll be looking through microscopes. Super, super, super thin, 7 nanometers. Still wider than a bit of DNA, but a lot smaller than some other structures, so it's thin. And then spanning it are these giant proteins. You can see you have the type of protein that goes right across the entire thing. This is called an integral protein, but you also have proteins that are not going the whole way across. They're only halfway across or just built into the one layer. So those are peripheral because they're on the edge. And then you have other proteins that are also on the edge, but maybe they have these little sugars coming off of them. Or they could have a fat with sugars coming off of them. If it's the sugar plus protein, it's a glycoprotein. If it's the sugar plus a piece of fat, it's a glycolipid. So those are your major structures within the cell membrane itself. You've got phospholipids, you've got integral proteins, or we also call these transmembrane because they cross the membrane. You have peripheral proteins, and you have your glycoproteins and your glycolipids. You also have cholesterol in there as well. Cholesterol is a type of fat, and it's going to be built in there as well to try to keep our membrane nice. You'll see why in a second. So let's get into the written details. One of the main things that we have to keep focus on is homeostasis. Cells have to keep the inside of their cell constant. So for temperature's sake, for how much water is in there, what the pH of that is, because we don't want to have it too acidic inside the cell and now the cell's enzymes don't work, or vice versa, too basic and now they don't work. So all those types of things have to be maintained to make sure that the cell's chemical reactions can keep happening. So you have to have what we call a steady state or homeostasis. The word homeo actually means same, and then the word stasis means like to stay put. So really this means same. You want to keep everything same. So you want pretty much constant conditions unless there's a reason to swing it in one way or the other. And we're going to do that not just on the big scale, which we'll study in our final unit in homeostasis, but you're also doing that right down on the cell scale, right on this micro level as well. There's 4 terms that you need to be comfortable with: permeable, semi-permeable, selectively permeable, impermeable. Really, these are just words that describe whether things can pass through the membrane or not. If the membrane is described as permeable, it can be permeable and allow one particular substance through, but at the same time, it can be impermeable to other things, so stops other things from going through. So if you look back up at our picture, you might want to have the gases like oxygen and carbon dioxide be able to go freely through. So you would say that the membrane is permeable to those things, but you certainly don't want the ribosomes on the inside to be leaving all the time. You don't want to lose your machinery on the inside. There's certain proteins and enzymes you want to keep inside. So you would want your cell membrane to be impermeable to those things, like a wall. We don't want anything escaping. Okay? Then the other 2 terms are also important. Semi-permeable means that if you meet the criteria, you're welcome to just walk through that door. So in other words, picture our classroom here. You've got a door on the side there. If that door is open and you are the right size and shape, you can fit through that door. So humans, easy. We can walk right through it. Mack truck, not so easy. Elephant, not going to make it. They're too big. So you just have to have the right characteristics yourself. Are you the right size? Because we're talking cellular level as well. Maybe it's a negative channel, but you are positively charged. Well, that's great. You'll be attracted to it and you'll be sucked right through. But if it's a negative channel and you are negatively charged, it's going to repel you. You can't make it. So the polarity is going to matter as well, et cetera, et cetera, et cetera. The charge and the polarity. Okay. So this is really it's an open hole. If you fit, you're welcome to come in. Selectively permeable has a little bit more to it than that. That's like we have an open door, but we have a bouncer there. So it's all ages night at the Rocks. Woohoo! And you got the bouncer saying, yep, no, you ugly, you can't come in. So there's a selective permeability that's happening there at the door with the bouncer. So you're going to regulate or control what's allowed to pass. Okay, so permeable, that particular substance can pass. Impermeable, no way it's getting in. It's like a shut-off wall. Semi-permeable, if you're the right size, right polarity, right charge, sure, come on in. If you're not, you can't. You can't physically make it through. And then selectively permeable, I'm going to decide when and who is allowed to enter and when and who is allowed to exit as well. Keep in mind, we're going both directions when we're transporting here. So we'll come back to that afterwards. On the right-hand side, we've got a much better diagram of what the actual phospholipid is supposed to look like. So I hope you recognize we've got a glycerol here. So we've got the 3 carbons and then the ester linkages that are happening here. 2 of those spaces are filled up with fatty acids. And then the third location, that oxygen is attached to a phosphate, which is attached to this big nitrogen group up here. This whole head, this section right here with the phosphate and the nitrogen, is what makes the head polar. And of course, we have these long nonpolar fatty acid chains. Overall, there's no charge because the carbon and the hydrogen are going to share electrons quite well. So what is the whole purpose? If I ask you what is the function of the cell membrane, you're going to give me these 3 things. I'm going to transport, but because you're a 12U student, you're going to be more specific. And you're going to say we're going to transport raw materials in, things like oxygen and glucose. And we're going to transport out anything that I've created for the benefit of other cells, so manufactured products, and any of the waste. So things like the ammonia, the urea, carbon dioxide is the waste from cellular respiration, etc. So that's what you're transporting. The function is also to prevent. I want to stop things coming in. I want to be impermeable to things I don't want coming in, and I want to be impermeable to things I don't want leaving. Again, you don't want to lose your ribosomes and your enzymes that are supposed to be inside, and you don't want foreign things coming in unnecessarily. It would just gum up the whole system of chemical reactions, all sorts of stuff, right? And then finally, it's communication identification. So saying, I am this type of cell, and telling other cells around, I'm healthy or I'm sick, please come and destroy me, whatever they need to say. I'm this type of cell that belongs to so-and-so. So let's get then into the actual parts that are going to help to contribute to transport and to the communication. So here again, we'll just talk briefly about the phospholipid bilayer. Bilayer meaning 2 layers, about 7 nanometers thick. You have a glycerol backbone with the 2 fatty acids in the phosphate group, like you see in the picture above. Remember that lipids don't dissolve in water because they are nonpolar, and that's going to form this barrier around the cell. The phosphate edges define and contain the fluid inside and separate it from the fluid outside. There are little tiny spaces, because these are physical objects, right? There are little tiny spaces that very small molecules are able to pass through. So things like oxygen and carbon dioxide can pass through freely because they're so small, and they're not worried about having any charge issues with being able to come through. If they were polar, they wouldn't be able to. So when we say here that water is allowed in, water doesn't really freely pass. It actually has these pores called aquaporins. Aqua meaning water, porin meaning pore, holes. So there are actual special things that allow the water to come in and out freely, little channels, because otherwise it is a polar molecule, and polar molecules can't pass freely. We would say that it would be lipid soluble, able to be dissolved in a liquid, so it would have to be nonpolar in order for it to pass freely. So fat soluble or lipid soluble molecules can also go in to a certain size. And then other little things like glucose, the urea, little amino acids, so building blocks of proteins that I need, they're going to have to be transported by those proteins we saw that were crossing the entire membrane. And then really large things, they might not be able to come in freely. I might have to actually pump them in one direction or another, and it'll require energy to do that. So for the most part, I'm actually going to use a process called diffusion to allow things to just flow. You got lots on the outside, great. If it can fit, it'll flow to the inside, just randomly colliding with things and flowing to the inside, spreading out. But if it's something that I need more often, I might have to purposely grab it from the outside and throw it to the inside. And in that case, I'm actively transporting it. I'm using energy to make sure I have enough of my supply. The membrane proteins are, again, the integral versus the peripheral. So integral, also called intermembrane, pass all the way through. Our 2 versions are channel and carrier proteins. I'll show you more detail on that in our next lesson on transport. And then peripheral are on the surface, so either on the inside surface or the outside surface. And they tend to be things like receptors. So they're going to, if they're on the outside, they would receive information from either a molecule or another cell, and they would change shape that causes a chemical reaction on the inside that can send information to the nucleus or to other parts inside. or they are called markers for the cell, and that is for cell recognition. So you learned about blood types back in grade 11. Type A, type B. Type AB has both A and B markers. Type O doesn't have any of them. Those are identifying that particular type of cell. And you know that if you are a type, let's say you're type A and you need a transfusion, if they give you type B blood, your body's going to say foreign, 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 foreign. It's going to attack that B blood, and you are in danger of dying. It could kill you if you have the wrong blood transfused. You don't want your immune system to be overwhelmed with all these foreign invaders that they can go and attack. And the reason they know to attack them is because they don't recognize the peripheral markers that are on the surface of the cell. They are scattered all the way, kind of like raisins in raisin bread or chocolate in a chocolate chip cookie. But they can move, which is why we say that it's the fluid mosaic model, because things can actually shift around. And you saw that in the animation earlier, where you saw the cholesterol rafts kind of stuck together, and they are free to move throughout the phospholipids. Then we get the glycolipids and the glycoproteins. Again, sugar added to a fat, sugar added to a protein that's buried in the membrane. And that carbohydrate chain is going to stick out, and again, it's going to be for communicating with other cells and also saying, hi, this is who I am, please don't attack me. Or maybe, yeah, I'm done, end of life, come and destroy me. Whatever it needs to say, it can send a message up. It could even say, I need help over here, send me this particular product. And then another, the messenger would come along and be like, oh, this guy needs something. Okay, and then it'll go and tell the message to the next cell to produce the product that can then flow through the bloodstream and come back to this cell and give it what it needs, etc. So they can talk to each other. The cells have their own communication system. Then we have the cytoskeleton. That's the microtubules and microfilaments that you saw in the video. They're going to be those little protein subunits that you saw forming and then being torn apart. And then you can add them here, move them there. You can do whatever you need to do with your cytoskeleton. It's supposed to give shape to the cell and also act as that transportation highway to be able to move things around, like the vesicle you saw with that little kinesin motor walking it around. Then we get cholesterol. Again, it's a type of fat. Cholesterol is built into the phospholipids, and it's going to have two very important things that it's going to do. Number one, it's going to make sure that the membrane doesn't just allow things to come through freely. So it makes it less permeable than it would be otherwise. We don't want things just floating in and out. We want some control over what comes in and out. And then the other thing that it's going to do is be able to make sure that when it gets really hot, that the fats don't turn into an oil and liquefy. And when it gets really cold, that it doesn't freeze. This is a test type of question. It'll be, basically you'll be telling me what the function of cholesterol is. I also want you to keep in mind that you make all the cholesterol that you will ever need. So you don't have to get it in your diet. That's why when people are having trouble with arteries hardening and then that's leading to heart attacks and you get blockages and all that sorts of stuff, maybe their coronary arteries are blocked and that's going to cause not enough oxygen to get to the heart tissue, and then heart tissue dies and then you have a heart attack, right? So cholesterol is involved with repairing the arteries. And so what ends up happening is when you get after tons and tons and tons of pressure, blood pressure being really, really high, begins to wear away at those endothelial cells on the inside of the blood vessels, creates little holes that have to be patched. So during the patching process, cholesterol often gets stuck in there and helps to form a plaque that begins to narrow the artery, which leads to further increase in blood pressure, which leads to further damage of the artery. Plus, if it narrows enough, you're not going to be able to get blood flow to the other side of that blockage. So the cells beyond that are going to die because they're not getting fed and they're not getting oxygen. So cholesterol in good amounts is going to actually help you. You need it for your cells. But if you have a high diet in cholesterol, it can lead to heart problems and heart issues. So here one more time, we've got our peripheral proteins that are on the edge. This one happens to be attached to a integral or intermembrane protein. Here's another one. You can see there's a channel in the middle there as it's been chopped open. Then you have the type of protein that can be attached to sugars, so it's a glycoprotein. Or if it's attached directly to a lipid, you can call it a glycolipid. Again, those are for communication. And then you have the phospholipid bilayer. Hydrophilic heads, they like water. Hydrophobic tails that go in. And then you have the parts of the cytoskeleton that are kind of just keeping everything stiff and together, and not so that they don't float away on each other. And of course, finally, cholesterol as well, built in order to strengthen that membrane. Make sure it's not so permeable, make sure it doesn't melt, make sure it doesn't freeze. And that, my friends, is the end of this lesson.